Let's see the title actually. The next talk is going to be given by Dr. Ray Wong, who's one of the scientists, senior scientists at the Pacific Heart, Lung, and Blood Institute, and he's been working on a variety. He's an immunologist by training and has worked actually in industry as well as at the Pacific Heart, Lung, and Blood Institute, and he's trained at USC and with a PhD in immunology, and he's started to look for last few years at strategies to enhance the immune-based therapies for mesothelioma. And I think he's got some pretty intriguing ideas and results. So, Dr. Wong.
Good afternoon, everyone. Certainly very exciting times for the field of cancer immunotherapy with the recent arrival of an entirely new class of cancer drugs called PD-1 immune checkpoint blockers, which we heard a little bit about this morning. A lot of press coverage on these new drugs that have just hit the market because many people, including myself, believe that we are potentially witnessing a paradigm shift in the way that cancer is treated. Now, immunotherapy, such as immune checkpoint blockers, don't treat tumors directly. They treat the patient's immune system and directs the immune system to do what it normally should be doing, which is detecting and destroying tumor cells. The pivotal point in this field was actually 4 years ago when the FDA approved the first immune checkpoint blocker, ipilimumab, for melanoma, but it's the second class of immune checkpoint blockers, the PD-1 blockers, that have generated even more excitement because of their apparent effectiveness across a wide variety of cancer types. There are two PD-1 blockers on the market right now, one called pembrolizumab and the other called nivolumab, which I actually worked on for my PhD thesis 10 years ago when I was at the USC Norris Cancer Center, back when the drug didn't have a name yet. It just had a pharmaceutical code name at the time. It was just recently synthesized, and it was still in the preclinical R&D phase that I was involved in as an academic collaborator. But now that the FDA has approved PD-1 blockers for treating melanoma and lung cancer, this has catalyzed the launch of numerous clinical trials testing their effectiveness in a wide variety of cancer types. Currently, there are at least 150 clinical trials testing either nivolumab or pembrolizumab for at least 30 different cancer types. At least 2 of those trials include pleural mesothelioma. So how do PD-1 blockers work at the molecular and cellular level? These are engineered antibodies that block an immunological receptor on T cells called programmed Death1 or PD-1, and this prevents its interaction with one of its natural ligands, PD-L1, which is often expressed on tumor tissue, including pleural mesothelioma. And the net effect of blocking this interaction is enhanced T cell activity and killing of tumor cells. As far as the clinical effects of PD-1 blockers, the primary effect is overall extended survival, or in other words, decreased chance of death for the average patient. There's tumor shrinkage in select cases, on the lower end, about 15 to 25% of treated patients. Some rare cases of complete remission. Responding patients often experience durable benefit that can sometimes last years, even after completion of treatment. And there's some evidence that reinduction therapy is also effective, meaning that some patients who initially respond to treatment, if they relapse while off treatment, they can be successfully retreated with the same PD-1 blocker without any apparent drug resistance. Pembrolizumab is now being tested in pleural mesothelioma patients, with the first preliminary data being presented just 2 weekends ago at the AACR conference, and that presentation can actually be viewed online for free at their website. But to summarize, 25 patients have been treated so far, and about a quarter of them have some evidence of tumor shrinkage. No complete remissions so far, but the trial is still ongoing. So what exactly is, or what likely is happening in the other 3/4 of patients who apparently aren't responding to treatment, and why are we seeing only partial tumor shrinkage and not complete tumor regression in the responding patients? Well, tumor tissue often harbors multiple immune resistance mechanisms, and this collectively creates a microenvironment that allows tumors to escape detection or otherwise complete eradication by the immune system. PD-1 blockers only address one particular immune resistance pathway, Some of the other major immune resistance mechanisms that are often present in tumors include the abnormally high presence of immunosuppressive cells, such as T regulatory cells and macrophage, myeloid-derived suppressor cells. Also high levels of certain proteins that are believed to suppress immune responses against tumor tissue. Now, conventional therapy might be effective at reversing some of these immune resistance mechanisms, and we actually published a paper last year discussing the major theories of how chemo, radiation, surgery, and even cryoablation each have potential for reversing some of these immune resistance mechanisms in pleural mesothelioma and potentially sensitize the tumor to more effective treatment with immunotherapy, such as PD-1 blockers. And there are now clinical trials being launched for a number of different cancer types where conventional treatment is being combined with PD-1 blockers to hopefully achieve better tumor responses, and I think this will eventually pave the way for its application to pleural mesothelioma. There are still some challenges regarding the effective combining of conventional treatment with immunotherapy, in that the optimal combination regimens have yet to be established. There's still unknowns regarding the best drug choice if we're talking about chemo, the optimal dosages of chemo, the optimal dosages of radiation, how much of the tumor to irradiate, and also the timing of each individual treatment modality. There's some limited data from patient case studies, in particular the combination of radiation plus immune checkpoint blockers that might provide some guidance. There are at least 3 patient case studies that I know of in the medical literature, 2 in melanoma and 1 in lung cancer. But given that the safety and tolerability of combining different treatment modalities have yet to be established in large patient populations, as far as experimental agents to reverse immune resistance of tumors, the approach that is garnering the most attention right now, especially in the pharmaceutical industry, is the use of experimental agents to block a particular enzyme called indoleamine 2,3-deoxygenase, or IDO. This enzyme, IDO, is often present in tumor tissue, and it catalyzes the local depletion of tryptophan, an essential amino acid that is important for optimal immune function against tumors. And these experimental agents targeting IDO are now also entering clinical trials and being combined with PD-1 blockers to hopefully achieve better tumor responses in a number of different cancer types. As far as preclinical exploratory approaches to reverse immune resistance of tumors, there are specific antibodies or toxins or modified toxins to selectively deplete certain populations of immunosuppressive cells. These are generally effective in animal tumor models, but haven't really gained much traction as far as pharmaceutical development for clinical implementation. What we've been working on at our lab is a provocative strategy to use a type of stem cell that has a natural attraction for damaged tissue and tumors. In animal tumor models, these cells, they're actually called mesenchymal stem cells. In animal tumor models, they appear to be effective drug delivery vectors for improving the targeting of anti-tumor agents towards tumor tissue. So this has led us to believe that human mesenchymal stem cells could potentially be effectively engineered to deliver immune-modulating agents that might reverse immune resistance of tumors, which in and of itself could be effective as a single-agent therapy, or it could serve to sensitize tumors to more effective treatment with immunotherapy, such as PD-1 blockers. So what exactly are mesenchymal stem cells? These are, their normal function in the body is to serve as a reservoir of cells to maintain and regenerate tissue, primarily fat, cartilage, and bone, but possibly also muscle, neurons, and blood vessels. So there's a lot of interest in these cells in regenerative medicine. For therapeutic purposes, they can be harvested from different tissues, including bone marrow, body fat, umbilical cord blood, and term placenta. The earliest reports of using mesenchymal stem cells to treat experimental tumors in animals goes back to at least the early 2000s, so well over 10 years now. The study that's most relevant to pleural mesothelioma was actually published just last year by a lung disease research group in the UK, where human mesothelioma cells are implanted into the chest cavities of mice to mimic the anatomical location of pleural meso in humans. The mice were then treated with mesenchymal stem cells engineered to deliver a tumor cytotoxic protein called TRAIL, and this resulted in significant shrinkage of established tumors in the chest cavity. An interesting observation in this particular study was that intravenous infusion of the therapeutic was more effective than instilling it directly into the chest cavity. But of course, mice and humans are different. The optimal route of administration for this new type of experimental therapy still needs to be evaluated and explored in clinical trials. But encouragingly enough, in Germany, there's a biotech company that recently completed a groundbreaking first-in-man phase 1 clinical trial using engineered mesenchymal stem cells to treat gastrointestinal cancers. It's a somewhat complex treatment in that it's personalized for each patient, where bone marrow is extracted from the cancer patients themselves. It's sent to a manufacturing facility. There's a 10-week turnaround time to manufacture the working therapeutic product, and then it's reinfused back into the patient. Now phase 2 clinical trials are underway, which is an encouraging sign that this new type of experimental therapy at least appears to be safe for use to humans. Its actual effectiveness against tumors is still unknown. That's usually a question that's answered in larger clinical trials. Now, one of the major issues for experimental mesenchymal stem cell therapy is the tissue source used to manufacture the final working therapeutic product. As I mentioned, the trials in Germany utilize bone marrow extraction from the cancer patients themselves and a 10-week turnaround time to manufacture the final working product for reinfusion back into the patient. Now, 10 weeks is a long time to wait, especially for a patient with an aggressive cancer like pleural meso. So there would be great value in a product that is pre-manufactured, basically an off-the-shelf therapy that would be available for immediate use. And such an off-the-shelf therapy would be manufactured from the tissue of healthy donors, whether it's bone marrow or body fat, cord blood or placenta. But the obvious issue there is tissue compatibility between genetically unrelated donors and recipients. But what we do know about the basic biology of mesenchymal stem cells is that they are not easily recognized as foreign tissue in the body. So these types of stem cells could very well be universally compatible, or at least not immediately rejected as foreign tissue by recipients. And there is such a product, an off-the-shelf, pre-manufactured mesenchymal stem cell therapy that is reportedly entering first-in-man clinical trials for cancer patients later on this year in Germany. So we'll be watching very closely, of course, and adapting our own research in parallel to help answer important biological questions that might be relevant towards optimizing the clinical safety and effectiveness of this new type of therapy. So in the meantime, we're conducting our own research to produce a potentially universally compatible mesenchymal stem cell therapeutic that might have potential for reversing immune resistance in tumors. We've started off working with human term placentas to harvest mesenchymal stem cells because for research purposes, they're more readily obtained for work in the lab compared to other tissues such as bone marrow or body fat, and it provides more raw tissue to work with than cord blood. But regardless of the tissue source, harvesting and producing mesenchymal stem cells goes through the same process. There's an initial digestion or separation phase, then after 2 to 3 weeks of expansion in specialized medium and clearing out unwanted cells, we obtain a purified population of mesenchymal stem cells that can be used immediately or frozen away for future use. After harvesting mesenchymal stem cells, there's an important quality control step. We use the same methods as clinical grade manufacturing, which involves immunofluorescent labeling of cell surface markers to verify correct identity and high purity with trace amounts of undesired contaminating cells. As far as engineering the mesenchymal stem cells to deliver cancer therapeutics, there's a couple of general approaches. We can engineer the cells to produce proteins that directly kill tumor cells, but we think that there's more or greater potential in engineering the cells to produce proteins that reverse immune resistance of tumors, either as a single agent therapy or potentially to sensitize tumors to more effective treatment with adjunctive immunotherapy. For producing a prototype therapeutic in the lab, we use a replication-deficient virus. It's actually the same virus that's being used to treat pleural mesothelioma patients in clinical trials, but we use the virus to engineer these mesenchymal stem cells to produce interferon alpha. And the ultimate goal is to evaluate the potency of these engineered mesenchymal stem cells in test tube models of human tumors, where tumor cells are grown in three-dimensional clusters and embedded in a matrix that simulates invasion into surrounding tissue. And it's a test model that's also highly modular. It allows us to carefully add or subtract individual components of the immune system, which would allow us to carefully analyze the effectiveness and also the mechanism of action of this new prototype therapeutic. Now, we can engineer the mesenchymal stem cells to produce pretty much any protein therapeutic that has anti-tumor potential. We're starting off with interferon alpha because it's already FDA approved for treating a number of different cancers. The currently available interferon alpha is injected systemically, so it goes all over the body, and very little of it actually reaches tumor tissue, and it's also fairly toxic, so this limits its effectiveness as a systemic therapy, and this is where we believe that mesenchymal stem cells might be useful for improving the targeting of interferon alpha to where it's needed most, which is tumor tissue. As far as the putative effects of interferon alpha in tumor tissue, it stimulates endogenous immunity against tumors. It can counteract the effect of immunosuppressive cells. There's some evidence that might even directly neutralize T regulatory cells, which are otherwise shielding tumors from immune destruction, and interferon alpha is also anti-angiogenic. And we actually have created a prototype therapeutic in the lab where mesenchymal stem cells harvested from human placentas are engineered to produce significant levels of interferon alpha measured here by immunofluorescent labeling of interferon alpha inside the cells. But much of the interferon alpha is actually secreted and released outwards towards the extracellular space. So we've actually reached a pivotal point in this project with the successful engineering of human mesenchymal stem cells to produce interferon alpha, which hypothetically could be used as a tumor treatment. And we've actually observed some other really intriguing characteristics about these engineered mesenchymal stem cells producing interferon alpha. I won't go into detail today because we're going to try to publish a paper on it, but these are findings that, to my knowledge, have ever been reported before. And the timing of our research, or the progress of our research is very timely, especially with clinical trials using engineered, potentially universally compatible mesenchymal stem cells entering clinical trials for cancer patients later on this year in Germany. So to summarize the key points I've discussed today, first off, immunotherapy, in particular the PD-1 immune checkpoint blockers, are rapidly moving forward as a new option for treating diverse cancers, including pleural mesothelioma. But despite these recent advances, there's still a significant proportion of cancer patients who do not respond to immune checkpoint blockers, at least 50% or more, depending on the cancer type. So, and even among the patients who do respond, complete tumor regression or disease remission is very rare. So there's a lot more that we can do in this area, and it's likely that reversing immune resistance locally in tumor tissue is likely the key to enhance tumor responses. And lastly, we believe that mesenchymal stem cells might be useful drug delivery vectors for reversing this inherent immune resistance in tumors. Acknowledgments. First off, Irina Yankulescu, who actually helped me start up the Pacific Mesothelioma Center research lab about 2.5 years ago, and established the mesenchymal stem cell research program literally from scratch. There was no pre-existing work to build off of, and now we have something of value that we can build on for the foreseeable future. She's since actually changed career fields. She's a medical writer now at Cedars-Sinai at Beverly Hills, just down the street from us. And of course, best wishes for her new endeavor. And our scientific advisors, all of whom are practicing oncologists or professors, Diana Gage, Olga Levsky, Warren Grunfuss, and Bob Cameron. Thank you.